To understand the genetic cause of Crouzon syndrome, you must first understand the basics of how human genetic information is stored and utilized. In the nucleus of all your cells, you have 46 chromosomes shown here. There are 22 pairs of chromosomes called autosomes labeled 1 to 22, and there are 2 sex chromosomes which are either XX in females or XY in males. The gene of interest which is related to Crouzon syndrome is located on chromosome 10. Let's zoom in on it. The gene which is related to Crouzon syndrome is called the Fibroblast Growth Factor Receptor 2 gene, which is shortened to FGFR2 for convenience. Its chromosomal locus or location is on the long arm called the Q arm of chromosome 10 in a position identified as 26.13. But what is the FGFR2 gene? Well, like all genes, it is made up of DNA, which is a long-stranded sequence of molecules called nucleotides. DNA is double-stranded, and hence the pairing of these nucleotides are called base pairs. You may have heard that DNA is called the biological blueprint, and this is because the sequence of nucleotides in your DNA gives instructions for how to make every protein in your body. Every gene is a sequence of 4 different nucleotides labeled as A, T, G, or C. The FGFR2 gene is 120,129 base pairs long. When FGFR2 protein is to be made, a messenger RNA or mRNA molecule must be synthesized by reading the FGFR2 gene in the DNA. This process, called transcription, is undertaken by a molecule called RNA polymerase. Many of the regions of this resultant transcript, called pre-mRNA, are not included in the final mRNA. Due to a process called splicing, regions of the pre-mRNA called introns are cut out and the remaining pieces called exons are glued back together. The FGFR2 gene contains 17 introns which are cut out during splicing and 18 exons which are glued back together. This is the resulting mRNA for the FGFR2 gene. It is worth noting that mRNA does not contain the T-base thymine and instead has a U-base uracil. This mRNA leaves the nucleus and heads to a ribosome, the site of protein synthesis, and is read in a process called translation. Groups of 3 nucleotides called codons on the mRNA correspond to a specific amino acid which is to be added to the growing peptide FGFR2. The mRNA has a 5' end and a 3' end and is read by the ribosome from 5' to 3'. Translation of the FGFR2 gene begins at a specific initiation codon, represented by AUG, and is terminated when a different codon called a stop codon with UGA is read. The region from the initiation codon to the stop codon is called the coding sequence, or CDS, and it is 2,466 nucleotides long for the FGFR2 gene. There are 12 nucleotides 5' of the start codon, called the 5' untranslated region, or UTR, and there are 770 nucleotides 3' of the stop codon, which makes up the 3' UTR. Overall, the translation process occurs to produce the functional FGFR2 protein.